Dr. Pinto, and thank you so much for the invitation. We feel blessed being here. These events really convey, it allows us to convey the messages from the parents, from the patients, from the physicians to keep our scientists motivated. So we are very happy to be here today. In contrast to Dr. Osland, I even need the help of a giraffe to say hello to you in Spanish. So a cordial hello from JCR Pharmaceuticals, and I was very happy to see that the color of the headphones was actually, it's the corporate color of JCR. We didn't sponsor it, but I'm happy to see this. So this is our disclaimer. Don't believe anything that we say. It's just a legal disclaimer, and we will also be disclosing some information about a drug called Itzcargo for the treatment of MPS II that is only approved for the treatment of MPS II in Japan. And a global clinical trial is ongoing. Who of you has ever heard about JCR Pharmaceuticals? 2 people, that's great. 3, 4, 5, wonderful. So we are a Japanese company. We are about 50 years in the business, very strong focus on rare diseases. We have subsidiaries also in the US, in Europe, in Brazil. US parents and individuals affected with lysosomal storage diseases, and US physicians, you are our heroes. Our problems are trivial against what you have to carry every day. We just switch off our laptop at night. We know that your problems are not switched off at night. And with our technologies and our programs, we work relentlessly to try to help you fulfill some of your hopes and dreams. Seriously. Dr. Osland did a wonderful job to tell you about the blood-brain barrier. I could ask the same question. Does anybody know what the blood-brain barrier is? It's a wonderful invention, actually. It prevents the passage of pathogens and toxins into the brain to damage the brain. The good thing, aspirin and alcohol can go through the blood-brain barrier, so that's a big relief. But the terrifying thing is that enzymes that are so important for lysosomal storage diseases to degrade the substrate, they go well into what we call somatic organs, but not so well into the brain. And this is why we have developed a very similar technology to Denali, the devil in the detail. I'm not going to bore you with all the science, but it has been over a decade of investment in deep research that finally resulted in the approval of Itzcargo for the treatment of MPS II, the somatic and the neurological symptoms. Something that we feel very, very humble about, and to see how it does change the life of individuals with MPS II in Japan. And we're trying to bring this innovation into the world. And I brought this medicine with me. This is Itzcargo. It's the antibody that binds to the receptor at the blood-brain barrier, gets transcytosed, and this red thing, the enzyme, is degrading the substrate in the brain. Is there anyone motivated adopting this little antibody? So whoever wants to adopt it. So how does it work? It crosses the blood-brain barrier, and as I said, it also goes into somatic tissues and organs. So you don't need a double treatment with the enzyme idosulfase and Itzcargo. It's really finding its way into the brain and somatic organs. It is expected, and we have seen this, the somatic disease is well controlled, and it provides additional benefit to the brain. What you see here is JCR's commitment in the MPS field, and I'll come to the other lysosomal storage diseases in a few minutes. JR141 is now in global phase 3 studies. We have a very similar design as Denali. We are in the US, in Europe, in South America. We are going to open many more sites also in other countries. I'll talk about this in a minute. MPS I, our drug, is currently in phase 1/2 studies, and we are committed to go into a pivotal trial next year, actually. We will go with JR441 actually in Europe in a phase 1/2 study also next year. MPS IIIB is a little bit later than MPS IIIA, and we hope that we will get this molecule into the clinic in 2024. And for the SLY syndrome, we don't know the timelines yet. I just want to tell you a little bit about the benefit of its cargo for the young patients, but also for those ones who have been with the disease for multiple years already. What you see here, I don't want to bore you with population data, but it's two siblings. The one in blue, he was the older sibling, diagnosed later, put on idursulfase, and what you see is the age over what we call the cognitive age, the age equivalent. He is now a little bit older. I mean, he's beyond 5 years and has the age-equivalent function of around about a 2-year-old boy. The younger brother had the luck of getting diagnosed earlier, and he was enrolled in its cargo trial very early. And what you see in green, we feel very humble how well this young patient is developing and not developing those progressive CNS signs and symptoms. But the message is, even individuals who have been with the disease for decades, and you see here individuals that were treated in Brazil with a more attenuated form of the disease between 25 and 45 years. And what you can see here, it's a very different scale. Treatment for just a year with its cargo even gave those individuals who have been long with the disease a very significant benefit for the executive, for the adaptive functioning, short-term memory, and many other aspects that holds them back to reach their full potential in life. Something that we also feel very happy about, that this drug provides hopes for young for the young patients and those ones who are with the disease for quite some time already. The trial design is something very similar to what Denali has done. We have the cohort A, that is the neuronopathic patients, and the cohort B, the more attenuated patients. My dear colleague Takayo Egawa is also with me today. She speaks much better Spanish than I do, and even than the giraffe does. So if you have any questions, feel free to reach out to us. We will be more than happy to tell you more about the program and the clinical study. What's important for you, the global phase 3 study has been going on for a while. We know the safety profile extremely well because this drug has been on the market in Japan, and one site that is already open in Spain is actually here in Barcelona. And we feel absolutely privileged to work with a physician here in Barcelona to provide this innovation to the patients who might be interested in enrolling in this study. I want to switch gears to our MPS I program because I feel this is also important for you to understand what is going on. These are the results from a phase 1/2 study, and we are measuring a biomarker of the substrate in the so-called cerebrospinal fluid. That's the fluid that goes around the brain and is an indicator. If you see a decrease, it's under certain circumstances an indicator that you also decrease the substrate directly in the brain. And what we see is a very good response in 100% of the patients. And we are very pleased with the safety profile, very pleased with the other clinical observations that we have seen in the study that is going on, not in Europe, unfortunately, but we want to bring this innovation also, of course, to Europe in our upcoming phase 3 study. We believe, if we look at the heterogeneity of MPS I, we believe that this drug that passes the blood-brain barrier can also provide significant relief to those individuals who are post-transplantation and may not live their full potential when it comes to the cognitive signs and symptoms. And it is our deep commitment to include this patient population or this subject population in our upcoming phase 3 trial as well. Again, we're happy to discuss details. JCR is unique in many ways. And I feel privileged that it's not always the business case that drives the decision of executive management, what we are doing. And what you see here is an ultra-rare disease portfolio. And it's sometimes very difficult to create a business case, a positive business case in this disease, but this is our commitment to many of the ultra-rare diseases. This morning I met the first individual with Focusidosis I've personally ever seen, Ruben, my little hero, and his mother, Vanessa. It was a very touching moment. She participated in our survey to even understand better Focusidosis. Our commitment is to go into clinical trials with this drug in 2025. I don't want to go through all those details, but our commitment, leaving no patient behind, goes beyond Focusidosis. This is what you see, what we can commit to as a company. We are limited in our resources, but we want to be held accountable to, not to our words, but to our deeds. Just real quick, Focusidosis, how do we assess this? This is really an ultra-rare disease, very difficult to address. I want to just show you some data in an animal model. So we modify the mice so that they have no enzyme anymore. We knock out the gene, very similar to what happens in individuals with Focusidosis. And what you see is the following. Normal mice, they have no substrate. When you have the knockout of the enzyme, you see the substrate is actually building up here in the plasma, in the kidney, in the brain, liver, spleen, and cerebrospinal fluid. And when you treat those animals with different doses of it's very similar to this one. If anybody wants to adopt another one, feel free to do so. It's very similar. The platform is very applicable to many antibodies and enzymes. And what you see here, that after 3 months of treatment, we do not only see a reduction of the substrate in what we call the peripheral organs, but also in the brain and the cerebrospinal fluid, which is a good indicator and gives us great confidence that those molecules hopefully will have some efficacy also in patients. When we look into the brain, I know you're probably only a few of you are scientists, but you see in the upper panel normal control, and then you see those white spots. We stained them for the activation of certain cells, and they should not be activated because this is a kind of an alarm signal. In the diseased animals, you see this alarm signal going up, and in the treated animals, you see this signal doesn't even come up. And what's also very important is, if we look at the cells in the brain, we don't want to get them destroyed. And what you see in the other part of this, in the lower part, this is a normal mouse, and here you see in the middle how many of those cells in the brain, the Purkinje cells, how they get destroyed by the disease. And this does not happen if we treat those animals with the antibody enzyme fusion protein. That's all I want to say to you today. Gracias. And again, if you have any questions, feel free to reach out to us. We would love to step into dialogue with you. Thank you so much.